For more than 400 years, the genetic story of Black Americans existed almost entirely outside the reach of hard evidence, passed down instead through fragments, a family name, a half-remembered story about a grandparent, and an unbridgeable gap where most family trees simply stopped, severed by the specific historical violence of the transatlantic slave trade, which stripped enslaved people of names, records, and any documented connection to the specific African communities they came from. For generations, that gap was permanent and, for most families, seemed genuinely unfillable. Then, geneticists combined DNA collected from more than 50,000 people on both sides of the Atlantic Ocean with centuries of surviving shipping records documenting the actual voyages of slave ships themselves, and for the first time, the two sources of evidence, one biological, one historical, lined up with each other in a way precise enough to reconstruct, region by region, exactly where the ancestors of millions of Black Americans actually came from, and exactly what happened to them once they arrived. To understand why this specific reconstruction took so long, and why it required combining genetics with archival history rather than genetics alone, you first have to understand the sheer scale and the sheer deliberate erasure built into the transatlantic slave trade itself. Between the early 16th century and the mid-19th century, traders operating under Portuguese, Spanish, British, French, Dutch, Danish, German, and Swedish flags forcibly transported more than 10 million African people across the Atlantic Ocean, drawn from an enormous stretch of the African continent, from Senegambia in the far west down through the Bight of Benin, the Bight of Biafra, West Central Africa, and Southeast Africa, regions as geographically distant from one another as Newport. Rhode Island is from Buenos Aires, on the receiving end of that same trade. This was never a single uniform migration from one African region to one American destination. It was an enormous, geographically dispersed, centuries-long system, and the historical documentation surrounding it, while genuinely extensive in certain respects, was deliberately structured in ways that erased individual identity almost entirely, recording enslaved people as cargo rather than as people with names, specific home villages, or family lineages worth preserving for their descendants. What historical researchers did manage to preserve painstakingly across decades of archival work was something considerably more useful for reconstructing the broader picture, even if it couldn't restore any single family's specific lost history. The Slave Voyages Project, a collaborative, multi-university research initiative, compiled detailed shipping records covering more than 36,000 documented individual slave ship voyages between 1514 and 1866, recording, wherever the surviving paperwork allowed, the specific African region a given voyage departed from and the specific American port where its human cargo was ultimately disembarked. This database represented an extraordinary historical resource on its own, but it could only describe the movement of ships. It couldn't, by itself, confirm what actually happened to the genetic legacy of the people forced onto those ships once they arrived, how their ancestry was ultimately distributed among the millions of descendants living across the Americas today, or whether the documented shipping routes actually matched the genetic patterns visible in modern populations. That is precisely the gap a team of researchers set out to close in a landmark study published in July 2020 in the American Journal of Human Genetics, led by population geneticist Stephen Micheletti, working with a large research team at the consumer genetics company 23andMe. The research team combined genetic data voluntarily contributed by more than 50,000 research participants on both sides of the Atlantic, people living across North, Central, and South America and the Caribbean, who carried more than 5% African genetic ancestry, alongside comparison populations carrying 95% or higher European ancestry and reference populations sampled from various regions across Africa itself, and directly cross-referenced this enormous genetic dataset against the historical shipping records preserved in the Slave Voyages database, As Micheletti described the research directly, the study combined genetic data from tens of thousands of people with historical records of enslaved individuals to construct one of the most comprehensive investigations of the transatlantic slave trade ever conducted using this specific combination of methods. The results confirmed, with a level of genetic precision no purely archival historical study could have achieved on its own, that the geographic patterns visible in modern African-descended populations across the Americas closely track the documented roots of the historical slave trade itself. Regions in the Americas that received the highest documented volume of enslaved people from specific African regions, according to the surviving shipping records, show correspondingly elevated genetic ancestry connected to those exact same African regions in the modern population living there today. As Micheletti stated directly regarding the study's findings, the genetic results were, in general, consistent with the shipping manifests and other historical documentation preserved from the slave trade era. A genuinely significant confirmation that these two entirely separate lines of evidence, one drawn from surviving paper records, one drawn directly from human DNA, told fundamentally the same underlying story. But the study's findings went considerably further than simply confirming known shipping routes. And this is where the genetic evidence began revealing details about the actual, lived experience of enslaved people that written records alone had left incomplete or entirely obscured. The research team found clear, direct evidence connecting specific regional patterns of African ancestry in the Americas to some of the darker, more deliberately suppressed methods used to control and exploit enslaved populations once they had actually disembarked. One particularly striking finding involved the documented extraordinarily high mortality rate among enslaved men specifically forced into labor on sugar plantations, especially across parts of the Caribbean, where the physical demands and brutal working conditions of sugar cultivation killed enslaved men at considerably higher rates than enslaved women working in the same broader colonial economy. This historical reality, already documented to some degree in surviving colonial records, left a directly detectable genetic signature in the modern population. In specific regions with this documented history, the genetic data shows a measurably lower than expected male genetic contribution from enslaved African men relative to the contribution from enslaved African women, a pattern directly consistent with enslaved men in these specific regions having, on average, fewer surviving descendants than enslaved women, precisely because so many were worked to death before having the opportunity to become ancestors of anyone at all. A separate and even more widely and consistently documented pattern merged when researchers examined the specific balance between African and European ancestry along the paternal versus maternal genetic lines across the Americas as a whole. This specific question had already been examined in earlier complementary research, including a major 2014 study published in the same journal, analyzing genetic data from 5,169 self-identified African Americans alongside more than 150,000 additional participants of other backgrounds, again drawing on the 23andMe research database. That earlier study documented what its own authors described directly as pervasive mixed ancestry and asymmetrical male and female ancestry contributions across every population group examined in the research. Among self-identified African Americans specifically, the study found an average European genetic ancestry contribution of approximately 24%, a genuinely substantial figure, alongside a smaller but consistently detected average Native American ancestry contribution of roughly right around 1%, itself a genuinely meaningful finding, since it provides direct genetic confirmation of documented but historically underexamined contact and intermarriage between enslaved and free black populations and indigenous American communities across multiple centuries of shared history in the Americas. What made this specific asymmetry genuinely significant, though, wasn't simply the overall percentage of European ancestry detected, but the specific pattern of how that European ancestry was actually distributed once researchers separated out paternal and maternal genetic contributions using the same Y chromosome and a mitochondrial DNA methodology applied in comparable research examining other mixed ancestry populations across the Americas. Across the overwhelming majority of regions studied, European genetic ancestry among African-descended populations in the Americas was found overwhelmingly more often on the paternal line than the maternal line, a genetic pattern directly and unambiguously consistent with the well-documented historical reality of the systematic, sustained sexual exploitation and rape of enslaved and free black women by white slaveholders and other white men throughout the entire period of slavery in the Americas. A genuinely brutal chapter of history that, until this kind of genetic research became available, had been documented primarily through slave narratives, legal records, and the extensive historical scholarship built around them, rather than through direct physical, population-wide biological confirmation. This specific genetic pattern is not unique to the history of African Americans specifically. Population geneticists have documented remarkably similar sex-biased admixture signatures in several other historical contexts shaped by conquest, colonization, and forced or coerced population contact elsewhere in the world, including, as one directly comparable example, the genetic legacy of Spanish colonization in Mexico, where a broadly similar pattern, incoming male ancestry concentrated on the paternal line, existing local ancestry concentrated on the maternal line, reflects its own separate but structurally comparable history of conquest and coercion. The 23andMe research team's broader 2020 study also identified genuine, historically informative regional variation in exactly which parts of Africa contributed most heavily to specific parts of the Americas, findings that added real, previously under-examined nuance to the historical record. The research specifically examined genetic connections tracing back to several of the African regions and ethnic groups most frequently referenced in surviving slave trade shipping records, while also noting directly that additional, currently understudied African source populations, including communities from Sierra Leone, Senegal, Guinea-Bissau, and Angola, represent genuinely promising directions for future research capable of filling in still more of the picture as additional genetic samples from these specific regions become available for comparison. This body of research also connects to a separate complementary line of genetic investigation examining a specific and genuinely important question about the broader African ancestral gene pool itself, one addressed directly in later research analyzing what happened to the overall genetic diversity of enslaved African populations once they were forcibly dispersed and mixed together across the Americas. Prior to the transatlantic slave trade, African populations across the continent's western and central regions carried genuinely substantial genetic differentiation between different regional groups, reflecting many thousands of years of distinct independent population history across an enormous and ecologically diverse continent. Researchers specifically investigating whether the massive geographically dispersed process of forced transportation and subsequent mixing in the Americas increased, maintained, or actually decreased this pre-existing level of genetic differentiation found genuine evidence of what researchers described directly as homogenization of the broader African gene pool in the Americas, meaning that populations originally drawn from genuinely distinct and differentiated African regions were, through the specific mechanics of the slave trade and subsequent generations of intermixing among enslaved communities themselves, gradually blended into a genetically less differentiated, more genuinely mixed African American population than any single one of the original African source populations had been on its own, A genuinely significant finding for understanding modern African American ancestry as its own distinct blended genetic story, rather than as a simple, unaltered transplant of any single specific African population or ethnic group. It's worth stepping back to consider directly why this specific body of research carries the weight it genuinely does, well beyond narrow scientific or genealogical curiosity alone. For millions of Black American families, the transatlantic slave trade didn't simply disrupt genealogical record-keeping the way ordinary historical migration or displacement might. It was specifically and deliberately structured through the practice of chattel slavery itself to actively strip enslaved people of their original names, their specific documented regional and ethnic origins, and, in the vast majority of cases, any surviving written record connecting them back to a specific African community, family, or homeland at all. This deliberate erasure left a genuinely unique kind of historical wound, one considerably harder to address through conventional genealogical research alone than the ordinary gaps and uncertainties that affect most family history research in other contexts. Direct genetic testing, particularly once combined rigorously with the kind of detailed historical shipping records the Slave Voyages project has painstakingly compiled and preserved, offers something genuinely unprecedented for many Black American families. A real scientifically grounded, statistically meaningful connection to specific African regions, filling in at least part of a gap that, for the majority of American history, had simply been treated as permanently, irretrievably lost. Rebunded Iweala, who has spoken publicly about the significance of this research and is himself Igbo, the child of Nigerian immigrants, described the emotional weight of this specific kind of genetic connection directly, noting that traveling throughout the United States, the Caribbean, and South America, he has repeatedly been struck by the long-standing, deeply rooted connections between people across the African diaspora and the African continent itself. Connections that reflect genuine resilience and cultural continuity even within a broader historical context defined by deliberate, sustained violence and erasure. He described this specific sense of familiarity as something he has felt in genuinely disparate places, from St. Simon's Island off the coast of Georgia to the streets of Jamaica to Bahia in Brazil, a feeling he compared directly to walking through the streets of the specific village in Nigeria his own family originally comes from. Put all of this research together, and the genetic story of Black Americans that emerges is considerably more specific, more historically precise, and in its own way more genuinely remarkable than the gap left by centuries of deliberate historical erasure ever allowed most families to fully understand about their own origins. Modern genetic analysis, combined directly and rigorously with painstakingly preserved historical shipping records documenting Kant's more than 36,000 individual slave voyages across more than 3 centuries, confirms that the regional distribution of African ancestry across the modern Americas closely and consistently tracks the actual documented roots of the transatlantic slave trade itself, connecting specific African regions to specific American destinations with a level of genetic precision no purely archival approach could have achieved working alone. Layered directly on top of that African ancestral foundation, the genetic record also preserves a real, statistically unambiguous, and historically damning signature of the specific forms of exploitation and violence enslaved people were actually subjected to, from the brutally elevated mortality rates suffered by enslaved men on Caribbean sugar plantations to the pervasive, systematic sexual exploitation of enslaved and free Black women that left an unmistakable, sex-biased genetic pattern still detectable across the entire African-descended population of the Americas today. More than a century and a half after slavery's formal legal end, None of this is a story genetics simply invented from nothing. It is the same story African American families, historians, and communities have been telling for generations, now independently confirmed, region by region, and in many cases, family by family, by the direct, physical, unambiguous evidence written permanently into the human genome itself.